Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapalli, Assistant Professor at Department of Pharmaceutical Science and Technology, Institute of Chemical Technology, Mumbai. So, in our previous lectures, we started with the types of nanocarrier systems. In that, we have finished with the polymer based nanocarrier systems. In our past lecture, we have discussed the lipid based nanocarrier systems. In that, we have completed liposomal formulation systems, as well as we have also learned the importance of solid lipid nanoparticle and their preparation methods. And also we have seen the what are the nanostructure lipid carriers. In our previous session, we have seen the introduction about nanostructure lipid carriers, and then we have seen the types and composition of nanostructure lipid carriers. Then further we have seen the various methods of preparation for nanostructure lipid carriers, and after that we have discussed the case study of the nanostructure lipid carriers. In my today's session, I am going to discuss on various vesicular drug delivery systems. In that, the one of the system we have already discussed, that is the liposomes. So, apart from that, we have the various vesicular drug delivery systems, which are niosomes, ethosomes, the transferosomes, and bilosomes, virosomes, springosomes, and then phytosomes. So, today we are going to discuss what are the various vesicular drug delivery systems. In that we will see the what are the components which are associated with the various vesicular systems, and then we will see how it works, what is the mechanism, what are the functions of these vesicular drug delivery systems, and then we will see how which is effective in as in drug delivery systems. So, we know we started the introduction on the nanocarrier systems, and in that the one segment comes, that is a vesicular drug delivery systems. It is a very big family. In that we started already with the liposomal formulations. So, here you can see the definition, what are the vesicular drug delivery systems. Vesicular drug delivery systems can be defined as highly ordered assemblies consisting of one or more concentric bilayers formed as a result of self-assembling amphiphilic building blocks in presence of water. So, now we will try to understand based on our previous learning. We know that we have seen in case of liposomes, even in case of solid lipid nanoparticles as well as nanostructure lipid carriers, we have seen these systems are self-assembling building blocks of the various phospholipids. So, here when you disperse such systems, such components in water in presence of surfactants, without surfactant, which may form self assembles. So, which are called as vesicular systems, also known as nanovesicles. So, this will help for the various drug delivery applications. So, these are the complete list you can see with the pictorial representation. We can see all these types of the vesicular systems, which we can use it for the drug delivery and other cosmeceutical and various applications. So, here you can see the niosomes, and the ethosomes, trans-ethosomes and the transferosomes, and there are the other vesicular systems, preferably bilosomes, cubosomes, proneosomes and the chitosomes and the phytosomes. In this, today we are going to focus on the few types of vesicular drug delivery systems. Now, we will start with the niosomes. We know at this moment what are the liposomes. Liposomes are bilayer self-assemble lipid vesicles. And we know that phospholipids have limitation in drug delivery in terms of storage, which may undergo oxidation and various degradation pathways during the storage, which is the one of the major limitation with the lipids. So, now these are the niosomes are the alternatives to the liposomes. So, what are that? Niosomes are bilayer vesicles composed of non-ionic surfactants which are biodegradable, relatively non-toxic, more stable and inexpensive, which are alternative to the liposomes. The name itself, you can by seeing that you will understand niosomes. Neo is nothing but non-ionic surfactants. Wherever zomes comes, that is nothing but it is a body, or you can say some structures which will help to carry the molecules. So, niosomes are bilayer vesicles which are composed of non-ionic surfactant. Among with that we can use the water and other active ingredients. So, which are biodegradable and non-toxic compared with the cationic, or you can say anionic such type of surfactants which are less in toxicity, because when you use ionic surfactants they may interact with the cell membranes and other cells, which leads to the toxicity. Yes, which are more stable and inexpensive compared with the lipids. So, the second point, the non-ionic surfactants formed in presence of cholesterol based aqueous media. So, similar to the liposomes, here also we can use non-ionic surfactant that must be dispersed in a cholesterol containing aqueous medium, need to apply some of the heat, or we need to provide some physical agitation. With that we will get niosomes. So, due to the presence of hydrophilic and the lipophilic moiety, you can see in this structure, this is the aqueous compartment in the interior portion, and between these layers, so you will see that is the hydrophobic portion. With that you can accommodate, you can load both hydrophilic and lipophilic molecules into this structure, which will enhance the solubility similar to the liposomes. Hence, these vesicles holds hydrophilic drugs within this aqueous compartment, and whereas hydrophobic drugs will be encapsulated in this bilayer structure. And with this you can see this is span 60 structure, this hydrophobic tail and this hydrophilic head. This structure is similar to the liposomes, except here we use the non-ionic surfactants. Now, niosomes are prepared from uncharged, this point is very important, which are nothing but non-ionic surfactants with the cholesterol, whereas liposomes are prepared from the double chain phospholipids, which if you use in a neutral, that is which will not give any charge, and if you use an ionic type of the lipids, which will give the negative charge, and there are the cases you can also use cationic based lipids, which will give the positive charge in the finished dosage form. Now, the concentration of cholesterol in the liposome is much more than in the niosomes, because we know that cholesterol has a property which will maintain the fluidity and it will maintain the rigidity in the structure of the liposomes, and that will stabilize the formulation of the liposomes. Whereas in the niosomes, we use non-ionic surfactant, it will not require much amount of the cholesterol. As a result, the drug entrapment efficiency of liposomes become lesser than the niosomes. Yes, because of the high amount of the cholesterol, which will hinder the encapsulation of your active molecule in the structure of the liposomes. Both niosomes, liposomes are same in the functionality, but phospholipids in the liposomes are unstable, undergo the oxidative degradation. So, that is why which are one of the major limitation of the liposomes. So, that is why scientists have developed the niosomes, which are alternative to the liposomes and other lipid vesicular systems. So, now here you can see the various examples. Here you can see the fatty alcohols, ethers, esters, black copolymers. These are the well known surfactant, non-ionic surfactant, and we know that polysorbates, twins and spans. Poloxamer is nothing but pluronic F68, which is the triblock copolymer. We can also use it as a non-ionic surfactant. The rest of the surfactants, we can also use it as an components in the niosomal preparations. So, now we know that as we seen in the definition, which are uncharged moieties, uncharged carriers. Now, we know if any system which not contain any charge, what happen, that leads to the aggregation, because of these are non-ionic, which will not contain any charge on their surface. That leads to the aggregation in the during the storage. So, to avoid this, we need to provide this system. In some other cases, we have to modify the surface by using various charge inducing molecules, either the positive charge molecules. In other cases, we can use negative charge carrying molecules, so which will reduce the aggregation. So, the particles that can be suspended in a final formulations. So, that is the greatest advantage when we add charge inducing molecules on the surface of the niosomes. So, based on the requirement, if your formulation undergoes the aggregation, we need to add charge inducing molecules. Now, here also this case also we can see, after modifying the charge, so the particle will not aggregate because of the repulsion between the two particles. So, that is why we need to use charge inducing molecules in the niosomal formulations. So, these charge molecules should be added to increase the stability of the niosomes by electrostatic interaction, which will prevent the aggregation and coalescence of the niosomes. So, if you want to add negative charge, you can use diacetyl phosphate DCP, or you can also use phosphatidic acid, which will provide a negative charge on your non-ionic surfactant carriers, which are also known as niosomal formulations. If you want to, if you are really looking to add on a positive charge on your surface, you can go with the any amine based components, that is a sterile amine, and also you can use sterile amine chloride, which contains both contain amine group in their structure, which will give the positive charge on the surface of your non-ionic surfactants. So, with this you can enhance the stability, and also you can control the aggregation and coalescence in your niosomal formulations. Now, these are the various preparation methods which will help to form the niosomes by using the non-ionic surfactants. So, these are the, as we have seen in the case of liposomals, similar type of methods we can see here. Ether injection method we have seen, film hydration method and the sonication method, reverse phase evaporation method, microfluidization techniques. So, you can refer our previous lecture on the liposomes. So, now how we will do, how we will develop by this general method. With this we can understand how we can prepare the stable niosomal preparations. So, now we can see here this is a non-ionic surfactant. As we have seen, it is a hydrophilic head and the hydrophobic tail. When you disperse in a water medium which contains the cholesterol, when you disperse, this will form the bilayers. So, self assembling in the hydration by this any of this technique, you can use ether injection or film hydration, which will give the bilayer vesicles. So, yes, you can load the red color representation. This is the in the bilayer structure. You can load hydrophobic drugs, and the blue color represents the hydrophilic drug. This is aqueous compartment. There you can load the your aqueous drugs or water soluble drugs. So, with this you can make both water soluble and water insoluble drugs for the preparation of the niosomal formulations. So, now coming to the another topic. So, ethosomes are the one of the mostly used formulation in case of the dermal application. Yes, so ethosomes are nothing but the name itself speaks that it contains ethanol in their structure. Ethanol based nanovesicles, which are also called as ethanolic based liposomes. So, here the extra component in the liposomal formulation, which are called as ethosomes. Ethosomes are lipid vesicles containing phospholipids, ethanol, water and the active components. So, this formulation is similar to the liposomes. Here we need to add the major proportion in this formulation is the ethanol and then water. This composition will help to penetrate the membranes, cell membranes of the skin. So, in upcoming slides we are going to see the function or mechanism of ethosomes. So, ethosomes are novel carrier systems which used for the delivery of the active components, which have the low permeability, nothing but low penetration through the biological membrane, mainly through the skin. We know that the outermost stratum corneum layer of the skin function as a principal barrier for the process of the transdermal absorption. Yes, if your drug is suffering with any type of the penetration and permeability, in such cases we have to use some of the penetration enhancers, so that your penetration enhancer will help to pass your drug molecules or some formulations through the cells. But without penetration enhancers also, you can use ethosomes or some other formulations which will also overcome the limitations of the low penetration drug molecules. Now, in the pictorial representation you can see, so this is the hydroalcoholic component, and this you can see the alcohol, and this the water and alcohol, that is nothing but ethanol. The remaining structure is similar to the liposomes, whereas in liposomes we will see only the aqueous compartment, but here you can see the combination of these two. So, now here the ethanol will help to pass through this stratum corneum. So, now we can see this is the outer side, this is the inner side. So, this is a barrier, the topmost layer of the skin. Now, if you give any molecule of which is a low penetration, for them is very difficult to pass through between these cells. So, but when you use ethanol, so the ethanol will disturb the stratum corneum lipid layer, then which will help for the easy permeability of the vesicles. So, these are the applications. Ethosomes can be used in the transdermal drug delivery. Also, we can use it as a skin whitening and other cosmeceutical applications. So, now here we will see the mechanism of ethosomes. So, mechanism of ethosomes can be classified into two steps. First one is the ethanolic effect. You can see in the picture representation action of ethanol, which is called as ethanolic or ethanol effect. So, what happens when you administer your ethosomes, which contain active molecules on the skin? So, it will interact with the lipid layers of the stratum corneum, and then that ethanol will disturb the stratum corneum lipid structure, that will help in the fluidity and the flexibility of the vesicles. Now, we will see in the pictorial representation. So, here you can see the action of ethanol. So, after administration, so these particles are the ethanol which will release from the carrier. So, that is the first step, that is interaction with the stratum corneum. Now, after that what happening, that ethanol will come out and interact with these lipid layers that will disturb the stratum corneum lipid structure. Now, you can see the arrangement of the polar heads, and you can also see the non-polar tails. Because of that ethanol which is disturbed their structure. So, in the second step, the disordered lipid structure will help in the fusion of the ethosomes. So, that is called as action of ethosomes. Now, you can see in here this structure is already disturbed. Now, once the ethanol effect is done, then the ethosomes will fuse into the structures of the lipid layer, and further it will enter into the deeper layers of the skins. So, that is called as the ethosomal effect. Now, we can read here. So, it is followed by the ethosome effect, involves fusion of ethosomes with the skin lipids, improving interlipid penetration and the permeation, thus resulting into drug release into the deeper layer of the skin. So, that is the idea behind the ethanol and ethosomal effect. So, definitely whenever you have a water insoluble drugs, such drugs you can disperse in ethanol, and that can be utilized in the ethosomal formulation, and then you can administer. With that you will see the enhancement in the permeation of the molecules. So, with this we will stop today's lecture. Thank you so much.